This video is part one of an exploration into protein structure determination. We're going to look at 4 different degradation reactions and see what they do to small proteins or proteins in general. We're going to start with looking at our octapeptide here as an example. The N-terminal residue of this octapeptide is isoleucine. It's the free ammonium terminal on the left, and the C-terminal is always on the right, and it is aspartate. Edman degradation, here's what's going to happen after we reveal. It's going to use phenylisothiocyanate to tell us what the amino terminal residue is. And what happens is this chemical cleaves the C-terminal bond of the isoleucine, which is the amino terminal residue, the N-terminal residue, and it cleaves it away from the rest of the protein, and the results would be these 2 fragments. 3-letter short forms and 1-letter short forms here, also off the handout. I don't know if you've noticed those. Okay, so that would be quickly telling us isoleucine is the amino or N-terminal residue. Cyanogen bromide clips the C-terminal side of the methionine residue, MET, except when there is a met-Cyr sequence, and I don't put met-Cyr sequences on tests. So in this one we have met with his, not a problem. It's going to cleave the bond here, giving us 2 fragments. Notice methionine has to be on the end because everything to the right of it got clipped off. And it's in red because that was on the end there. You're going to see the red at the end of all these next examples. Alright. Oh, one other thing you learned. This chunk has to be the C-terminal side because met kept everything to the left, which is the N-terminal side. Trypsin. Trypsin cleaves the C-terminal side of the alkyl ammonium or iminium. That's not spelled correctly. I'll fix that. Of lysine and arginine. Those are the 2 residues that have a carbon portion in the side chain that's of the alkyl variety. That's this one. And then a nitrogen with a positive charge. C-terminal of that gets clipped, and we reveal everything to the left of lysine is still with lysine, and everything to the right, and because there's only 1 amino acid over there, it's the C-terminal. Trypsin doesn't necessarily give us the C-terminal. There could have been many more in the chain over here and just be the big long fragment over there. Okay, chymotrypsin. Chymotrypsin is going to catalyze the cleavage of the C-terminal side of the benzene-containing side chains. There's 3 amino acids with benzene in them. Phenylalanine. Phenylalanine is this one. Tryptophan, not appearing on this tetrapeptide, but tyrosine is right here. Parahydroxyphenylmethyl group there. Chymotrypsin is going to cleave C-terminal of each, giving us 3 fragments this time. And there they are. They're in red when they're at the end of the fragment. That's the C-terminal side of each fragment because everything to the right of that got clipped off. This doesn't have phenols in any of them, so that must be the C-terminal piece. All those little sentences I'm saying at the end are leading towards the more important second video, which gives us these fragments after these experiments and then asks us what the sequence was to begin with. So I look forward to that video, and I hope this one on what happens with these digestive reactions to proteins, what happens to those. And I look forward to a next video.